for accepting our kind invitation. And we were supposed to invite her to come to Mexico City. Of course, it's not possible, but we hope that next year, or maybe whenever she can visit us, she will do it if possible. And for me, it's a real pleasure, because I think she's a very kind of inspirational figure, especially for young people and for women in science. And I will read a little bit about her, because she's a very, very well-known scientist. She's a rising star in the international science. So in fact, this is not her first time in Mexico. Well, this is virtually in Mexico, but I met here a few years ago because we organized a session on rigidity in the Material Research Society meeting in Cancun that we hold each year. So Lisa Manning is a professor of physics at Syracuse University and founding director of BioInspired Syracuse Institute for Material and Living Systems. She's an award-winning interdisciplinary scientist studying the mechanical properties of biological tissues and the failure of disordered materials. Her work to understand how the global mechanics of tissues impact cell migration and pattern formation provides new insight into embryonic development, wound healing, and cancer. Her work to understand fundamental excitations in disordered solids generates better predictive models for flow and failure in material from gases to earthquake faults. She earned her BS in physics and BA in mathematics from the University of Virginia in 2002 before attending graduate school at USC, University of California at Santa Barbara, when she earned a PhD in physics in 2008, advised by John Carlson and James Langer. She works as a postdoctoral fellow at Princeton Center for Theoretical Science from 2008 until she joined faculty at Syracuse University in 2011. Professor Manning has given over 100 invited talks and published 49 peer-reviewed articles. She was recently highlighted as one of the Science News top 10 scientists under 40 to watch, and has received additional honors and awards, including the 2018 Maria Gebb-Meyer Award from the American Physical Society, the 2016 International Union of Pure and Applied Physics Young International Prize, a Simons Investigator Award, a Sloan Fellowship, a Scialog Award, as well as several teaching awards. And also, I think it's not here, but she's also part of the initiative that is called STEM, or something, which is an initiative to bring people into science in the United States. After I read her bio, I went to the APS and I read the report about the status of physical science in the United States and all the. I was very much surprised by this report, very interesting. I encourage everyone to really take a look, because I mean, it's very, very interesting, the diagnosis of the situation. So thank you very much, Lisa. We are very happy to have you. It's really a pleasure, and I think it's going to be a wonderful talk as usual.
Thank you so much for that really kind and embarrassing, slightly introduction. I appreciate it very much, and I'm delighted to be here. And I just would agree with you that there's some really interesting reports out there about the status of women in physics, and also the status of underrepresented minorities in physics in North America. And so I think it's really something that we can all work together on. So I think you can see my screen. Is that okay? And everyone can hear me, I think. Okay, okay. Raise your hand or something if there's any technical issues. And so today I'm going to tell you about some of our work on biology. We also do work on glass physics, but I'm going to focus on the biology part of what we do today. And the title of my talk, unfortunately or fortunately, I feel is a little grandiose, but I think it's true. So I'm going to tell you why. So I think biological tissues, the ones that make up our bodies, can be thought of as mechanical metamaterials. And I'll try to make that argument today. So what you're looking at on this first slide on the left is a microscope image of collagen, which is a fiber in our bodies that makes up a lot of our body, and I'll talk about that. And then this image is the motion of a large group of cells in a culture that are taken from the lungs of human patients. And so we're going to be trying to describe the collective emergent mechanical behavior of these systems. Okay, before I get started on what we actually did, I need to highlight the people who did the work. So this is my group, and there's some folks in my group. A lot of the work I'm going to be highlighting today was spearheaded by a fantastic postdoctoral associate in my group, Ojan Damavandi, in collaboration with Varda Haig, and who's another postdoc, and Chris Santangelo. So the first question of the seminar is, are you a solid or a fluid? And if this wasn't a Zoom talk and we were all in the same place, you would be shouting out solid or fluid. So think for a second what you would shout. Okay, and some of you would shout fluid, and that answer is, of course, correct, because our bodies are filled with blood, right, and other fluids, and that's what makes up our body. But I think if you think about the sort of what you typically think is your physical corporal body, you think about that as the solid, you know, doing things like kicking a football or holding a baby. You have to support shear stresses and basically be solid-like in order to execute those functions. But I want to highlight that even your physical corporal body doesn't always behave like a solid. In processes like development or wound healing, the cells may have to move over long distances and change the global shape of the tissue. So now I'm going to show you some of my favorite examples. So this is an image, or a video rather, of a zebrafish embryo. And as I play it, you'll see that this round part here is the yolk. And these cells that are dividing and dividing on top are what is going to become the zebrafish embryo. And at a certain stage, the cells start moving down the sides of the embryo and flowing. And eventually you can see that that flow is necessary to generate the body axis of the zebrafish and generate a nice adult zebrafish. And a collaborator of mine, Eva Maria Schatz-Collins, took this beautiful video that I'm going to show you on the right. Before I play it, I want to tell you what it is. So these red objects are the nuclei of the cells. And it's right at that stage of the embryo where all of the cells are about to move down the sides of the embryo. And although the nuclei are stained, the cell bodies are not stained and they're transparent. So you want to envision this as a material that takes up all of the space, even though you're just seeing the nuclei part of it. You're just seeing the nuclei. Okay, so when I play this movie, so I think, at least to my eye, this looks very much like a flowing fluid. I don't know if it does to your eye or not, but clearly there's a big macroscopic flow and the cells are jostling past one another and rearranging a bit, just like you might expect in a fluid. And so actually there was a recent, well, actually it's 2 years old now, paper out of Oche Campas's lab, who really looked carefully at the tissue fluidity as this body axis is elongating, as this tail bud is elongating. And what they showed is actually there's a gradient in the behavior. So at the bottom of the tail, the tissue is very fluid-like. And at the top of the tail, the tissue is very solid-like. And there's a clear monotonic gradient across the tissue. Moreover, that gradient is actually really important to the functioning of that tissue. It needs to be that way so that the thing can grow and then solidify and grow and solidify and grow. And so I'll come back to this a little bit later in the talk to talk about the mechanisms that are driving this gradient in the behavior of the tissue. Okay, so we saw that groups of cells, like with a with a lot of cell-cell contacts can behave either like a fluid. Sorry, just a moment. Can behave either like a fluid or a solid. And similarly, if we're looking at a fiber network, which actually composes large parts of your body. So things like the cartilage in your knee, for example, is made up of a lot of extracellular matrix. So there are no cells, but the cells secrete these fibers that align into these networks. And the interesting thing there is also a floppy to rigid transition in those networks. And so this is some work by Fred McIntosh's group and Heija Kondering's group, so that you can take a chunk of this collagen network. So you take this material and you strain it. And so that's what this axis is showing. If you the way it forms naturally in biological systems, the material starts out very floppy. And then if you pull on it or strain the system, there's a critical point at which it becomes rigid. So if you strain it enough, it stiffens and becomes rigid. And so this extreme strain stiffening behavior is another type of floppy to rigid transition. And finally, interactions between these two types of transitions could be important. So it turns out that a lot of recent work on cancer and invasion of cancer tumors studies the interactions between groups of cells or droplets of cells or a spheroid of cells embedded in extracellular matrix. And so as this matrix becomes stiff, it drives the cells to move out and invade the tissue. So there's these interactions between these two types of systems that play a key role in some diseases. Okay, and so we've actually written a paper that I won't talk about at all about what happens when you couple these two systems, like a group of cells and an extracellular matrix. But I won't focus on that today. Instead, I'm going to focus on what happens during this rigidity transition. And so we'll take this kind of fluffy, big picture question and make it a little bit more precise. And so the precise question we want to ask is, how do mechanical interactions between cells or fibers or cells and fibers dictate the emergent mechanics of a tissue? Okay. And so I wanted to highlight this quote by Phil Anderson. I feel that many condensed matter talks have to have a quote by Phil Anderson in them, so I will do that too. But I like this one because I think it highlights that although we take rigidity for granted, it's absolutely completely surprising that it exists. You push on a ruler and you expect the other end of the ruler to move exactly the same amount you pushed one end, right? But why is that? It is not stated in any fundamental law of nature. It's an emergent behavior. And I want to also highlight that, of course, that means that understanding it can be difficult, even in the case where the model is very simple. And my favorite simple example is sand. And so I imagine that you all have at one point in your lives taken a walk along a beach, walk on the beach. And so clearly you're walking on the sand, which means that it's behaving like a solid. It's supporting your stress. It's supporting a shear stress so that you can walk across the surface. But then, well, as long as you're not too close to the ocean, you can pick up the sand and sift it through your fingers. And that behavior is almost identical as you would expect from a fluid. And clearly not much changed between the time it was under your feet and the time it was in your hands. There's a very small change in density, as it turns out, but not much. And this other picture on the left is a pile of discs. And these discs have been manufactured to be essentially perfectly elastic. So it's the simplest thing you can imagine, a perfectly elastic, perfectly spherical disc. And the point that and they happen to be photoelastic, which means that these lines that you're seeing in this pile of sand are the force chains, the chains of forces that are running through the sand pile. And so I think this one beautiful picture highlights that even though the constituent objects are trivial as we can make them, they're elastic disks, the emergent behavior here is clearly complicated and difficult to describe. And there's many other examples. I happen to like sand, but in almost any physical field, and in biology too, this problem exists. But as a physicist, there is a silver lining. And the silver lining, I think, is that sometimes if you look at the statistics over very large numbers of objects, some of the features can become universal or be universal. In other words, the results that you get are not too dependent on the details of the model. And that's what gives us hope, because when I started thinking about this problem, about cells, because a single biological cell is an exceedingly complicated object, right? So if we have trouble answering this question about emergent behavior for an elastic disk, how are we ever going to answer this question about something as complicated as a cell? But then I decided as a physicist, I would try anyway, because maybe we could look for these universal features. Okay, so the question that's been really driving me crazy, really, for the past 5 years is, what are the physical mechanisms that give rise to this emergent rigidity in biological tissues? And the reason it's been driving me crazy is that it is not the answer I thought it would be. So I think the answer, and I'm going to try to explain it to you in the rest of the talk today, I think the answer is that biological tissues are behaving as metamaterials in a very specific way I will explain in the rest of the talk. What that means is that they acquire rigidity differently from the standard examples in physics. So by standard materials, I mean molecular materials like solid state physics, crystals, glasses, and also granular materials like that sand pile that we just described. And in order to describe this new type of rigidity, we need to develop a new theory. And so I'll talk a little bit about this brand new theory of rigidity that we just posted on the archive actually less than a week ago. So I'll try to walk you through it today. All right. So first, let's take the standard example again and talk about it a little bit more precisely. So if you are in an introductory physics class, how are you told, like when you think about how do I take something that is a fluid and turn it into a solid? How do I do that? Well, I either put it in a freezer, I change the temperature of the material, or I change the pressure, right? We all know that. That's one of the first things you learn, right, in physics or chemistry classes. Pressure or temperature. And so I want to talk a little bit about the pressure axis in particular. We all have an intuition that materials solidify as they get more crowded, as the packing fraction or the number density increases. So packing fraction is just the space taken up by the particles divided by the total volume of the box, right? So as things get more crowded, you solidify. Well, why in general? Why is that a very generic intuition? Well, I'll again highlight how it works in this specific simple example of jammed spheres. So disordered jammed spheres, and we'll take the limit that you go to 0 temperature, by which I just mean that thermal fluctuations don't move the particles around, just like sand. So if they're big enough like sand, then you don't have to worry about temperature. And in that case, well, how many degrees of freedom are there in a, let's say a jar of marbles? So how many degrees of freedom? Well, let's say there are NP particles or marbles, and each one of course has d degrees of freedom per particle, right? Because it can move up, down, left, right, side, right? Like, so there are NP, the number of particles, times D, the number of dimensions, degrees of freedom, ways that the constituent objects can move. And then there are, as I get more crowded, the particles start to hit one another. And each time they have a contact, that introduces a constraint on where the particles can be. So if each particle has little z, or z number of contacts, then the total number of constraints in the problem is just the number of particles times z, the number of contacts per particle, and then I have to divide by 2, and I have to divide by 2 because each bond is shared by 2 particles. Okay, so that means there are this many degrees of freedom and this many constraints. And then in mean field, you expect the system to become rigid when the number of constraints equals the number of degrees of freedom, right? And that happens precisely with just a tiny bit of math. It happens when the number of contacts per particle is exactly 2 times the number of dimensions, right? So at low densities, when I'm not very crowded, this coordination number per particle is less than the critical number, and the system is under constrained. And at high densities, this critical number is greater, the number of contacts per particle is greater than this critical number, and the system is over constrained and rigid. Cool. So somewhat surprisingly, I would say, this simple mean field argument works really well for a jar of marbles. You can actually do that, or it was first studied in a jar of peas. But anyway, it works very well. And it also gives rise to a lot of interesting behavior, such as topologically protected excitations and materials and things like this. So this works. There is one important extension, and therefore I'm going to go through a little bit of the details of this because it will be important later. So imagine now that I have a bar hinge framework. So now, and let's make it in 2 dimensions so it's easy to draw on a screen. Okay, and so here I have a 2-dimensional system and I have 4 hinges. So that is 2 times 4 is 8 degrees of freedom. I have 4 bars, so that's 4 constraints. And I have, in 2D, I have 3 rigid body motions, right? Translation in one direction, in another direction, and rotation. And so that suggests that I should have one non-trivial floppy mode, and the system is under constrained. And of course, I can fix that floppy mode by just adding one more constraint, and then I, again, have a rigid system, okay? But there's something I could have done that was a little stupid. So if I took these 2 plaquettes, these 2 squares, I could have put a rigid bar on each of them, and then I would have had a totally rigid system. But I could have placed the second bar in the wrong box. And then now I have a system where I have one part of my system that's rigid, but I have another floppy mode. And so if I want to count correctly, I have to count what's called these states of self-stress. So a state of self-stress is a pair of, or a set of forces on the system where the system still remains in mechanical equilibrium. So you can see now that whenever I put a redundant constraint, I get a state of self-stress. And so what this equation now says is, well, to be a little bit more precise, I not only have to know the number of degrees of freedom, I also have to, and so this is the degrees of freedom and the number of constraints. This is the number of 0 modes, and I should subtract these numbers of states of self-stress. Okay, so that's a little bit of an extension of constraint counting. But even with this extension, this theory does not explain rigidity in many under-constrained systems. So let me give you the example of a guitar string. So all of you know that if I have a guitar string and I hold the 2 ends of the guitar string between my fingers, and I hold them together at a length that is less than the length of the string, then the whole thing is floppy, right? And so now let's just to fix ideas, let's assume that that guitar string is made up of segments that are all springs, and that each individual small spring has a rest length, which I'll call L0. Okay, and so the game we're playing is asking whether do I hold my hands or the ends of the string closer together than all of the rest lengths of all of the springs, or farther apart than all of the rest lengths of the springs. And so if I hold my hands closer together, then the whole thing is floppy. But you can see that there's a critical point, and that critical point is precisely when I stretch my hands so it's exactly equal to the length of the string with no stresses in the string, right? And that at that point, it will become rigid. And if I keep stretching it, it will become stiffer and stiffer, and I can hear that stiffness by plucking it and hearing the pitch, right? That's how a guitar works, guitar tuning works. Okay, and so we saw that there was a rigidity transition from floppy to stiff at a critical point that doesn't depend on anything like the number of nearest neighbors or the crowding, right, in the system. We tune a single tuning parameter, and there's one state of self-stress that appears in the system at the critical point. And I just want to say that because I will be doing this for the rest of the talk, I talked about it as if I was changing the sort of the distance between my two hands, right? This big L, this length of the box. But I could have fixed the length of the box and just changed the rest lengths of the springs, right? That should have worked equally well. Those two things are equivalent. So I can either change the box or I can change the inherent length scale in the springs. Okay. So now, because I promised you a talk that talked about mechanical metamaterials, so I need to define what that is. And I think origami is a very good example of one. I'm not going to go through the details in a colloquium. There's a very nice Physical Review X paper that discusses this in detail. But the point is that this paper and several others have shown that origami becomes rigid due to a mechanism that is very similar to that of the guitar string. And again, in the colloquium, I'm not going to go through all the details, but I did want to define what I mean by mechanical metamaterial. So origami is an example. There's a lot of other examples. So I'm going to play a video by my friend Itai Cohen, because I think he does a better job than I do of defining what a mechanical metamaterial is. Let me play this video for you. And I think, can you give me a thumbs up if you can hear it once it starts playing?
The particular paper that we wrote in Science deals with the Mura-Ora fold. This is basically a series of parallelograms that, when folded into a sheet of paper, give that paper some mechanical properties that are determined by the fold patterns that we put in to the paper itself. The nice thing about this strategy for giving materials mechanical properties is that by changing the fold patterns, for example, I can give the paper a different effective stiffness. I can also introduce new novel mechanical properties. For example, if I take a banana and I just squish it in my hand, that banana is going to squirt out the other ends. But this particular pattern, if I expand it in one direction, expands in the other direction. And if I contract it, it contracts in the other direction.
Okay, so I think that's a reasonable working definition of a metamaterial. It's a material where the structure of the network or some large scale pattern that you put into it, that pattern or that network is what gives the material its properties, such as the folds in the origami, right? The folded origami has a mechanical response that is quite different from the mechanical response of the paper that makes it up, right? So that's what I mean by mechanical metamaterial. Okay, so the next point is that this is exactly what happens in these biological tissues that I introduced in the introduction. So let me walk you through that. So fiber networks can rigidify by tuning, well, the strain of the box. So I actually think you should start thinking about fiber networks as basically two-dimensional guitar strings, in the sense that they can start out floppy, and then if you just strain the box enough, all of a sudden they'll develop rigidity. And that's what you're seeing in this plot. So this is the shear modulus, a measure of the stiffness, as a function of the box size, how much you've strained the box. And this is a logarithmic plot. So what you can see is that for a while, they're pretty floppy. And then there's a characteristic strain at which some of them can stiffen by more than 3 orders of magnitude, right? And so this is a floppy to stiff transition. Okay, and this is we can often approximate and explain the behavior of these networks using a simple spring network model, basically just a 2-dimensional version of that guitar string. So there's a rest length, L0, for every spring in the network, and then you just pay a quadratic penalty for going away from that rest length. It's a very simple model, but it works well for these structures. And also those tissues that I talked about in the beginning, which I call confluent. So confluent means that there's no gaps or overlaps between the cells and the tissue. So all of the cells are touching other cells and they're just moving around and changing neighbors. So we have shown, and this is like a bunch of our work over the past 5 years in my group, that these tissues tune their shape. Okay, and I'll explain exactly how to measure that shape in a moment. But for now, I want to say that if I have a large shape index, I'm very elongated. And if I have a small shape index, I'm very rounded up. And so what this data is showing you is that if the cell shape is very elongated, then cells can change neighbors and behave very much like a fluid. And if the cells are more rounded up, then the cells don't change neighbors and they behave much more like a solid. So just like in the spring network, the spring rest length was this continuous tuning parameter. In cells, this shape is a continuous tuning parameter. And in case there's anybody that does a lot of biophysics in the audience, I just wanted to tell you, we've checked this now. This is brand new work. It very carefully, quantitatively in the fruit fly axis. So what we tell you to do is you measure this shape index, which is how elongated the cells are. And then you also, it turns out, have to measure the shape alignment, which is how aligned the long axis of the cells are. And if you do that, then our theory makes this prediction of this black line. And the important thing here is this black line has no fit parameters. So we wrote down this black line from pure thought, theory alone. Okay, and then we looked at data on cell shapes in the extending fruit fly axis, and we measured the cell shapes in a prescribed way that the theory tells you how to do. And then we looked at how fast cells were changing neighbors. Again, in a solid, you expect them not to change neighbors at all. And in a fluid, they change neighbors rapidly. And if you look at a single embryo, what you see is that the cell shapes change in this way over developmental time. And there's this critical point at which the system turns into a fluid. And it turns into a fluid right basically at the point that was predicted with no fit parameters by this theory. And if you look at lots and lots of embryos, again, you just fill in this phase diagram. I still find this remarkable, right? It's fluid here and solid here. And all you did was measure features of a snapshot of a fruit fly, right? You took one picture of a fruit fly and you could predict all of the dynamics in this fruit fly for the next 30 minutes, like whether the system is going to behave like a fluid or a solid. That's cool. Okay. How did we come up with this idea of this shape index? We used what's called these vertex models for epithelial layers. And in a physics colloquium, I can't describe all of the reasons that this is a reasonable model for these cells. So to a physics audience, I can tell you something simple. We think that cells control their cross-sectional area. So there's a preferred cross-sectional area and that there's a preferred cross-sectional perimeter. So again, you're looking at a cell, and if you look at a cross-section, it looks like a polygon in one of these confluent tissues. And so you can measure the cross-sectional area and the cross-sectional perimeter And there's a lot of biology that regulates that. And that's most of what I'll say about biology for this part of this talk. But as a physicist, you can appreciate that then if this is the sort of target perimeter and target area, well, the lowest order Taylor expansion is going to be quadratic in perturbations away from that area and that perimeter. So this is the simplest model you could write down, is that the cells pay an energy penalty that's quadratic if they deviate from that area or that perimeter. Okay, and then obviously if you have a model like that, then the parameter that you care about is precisely this target shape index, which is a cell's cross-sectional perimeter divided by the square root of the cross-sectional area, right? That's the simplest dimensionless number that you can write down. And it turns out that it dominates the physics of this entire problem. So again, I could give a whole colloquium on that and the biology of that, but that's not the point of this talk. I can answer questions, of course, about it. Okay, so so that is biology, and I couldn't help myself. I don't know how many of you have ever watched this movie, but I think that if you're trying to think about making machines or robots or something to accomplish tasks, then having materials that can transition on command between fluid-like and solid-like can really give rise to a pretty amazing set of behaviors. So for those of you who know, this is a scene from Terminator 2, and this is the evil Terminator 2, who clearly is transitioning between a fluid-like and a solid-like state in order to accomplish, well, in this case, evil tasks, but important tasks nonetheless. And so I think that when you think about what unites all of those biological systems, it's the fact that they just tune a single tuning parameter. And by tuning that single tuning parameter, they don't have to change the topology of the network. They just change that internal parameter and they can go from rigid to solid and back again. So if we can understand how this works, then you could start to design materials that have these properties too. Okay, so now I have to sort of go through a little bit of math, not too much math, but the idea is that we wanted to describe this type of rigidity, and we need to go beyond constraint counting. And so we wrote what I call our rigidity manifesto. It's kind of a long paper. We just posted it on the archive a few days ago, actually. So you're one of the first people that gets to hear really about this work, and you can actually go look at it if you want to on the archive. So let me tell you what it is. So how do we describe rigidity? Well, there's a huge history, especially in mathematics and amongst structural engineers, people building bridges and things, to explain when something becomes rigid. So this is the old answer. You focus on the geometry of a structure. So maybe some of you have heard about tensegrity frameworks. There's an old idea actually that this is a good model for cells, which I think is valid. But tensegrity frameworks are ones that have bars and hinges, but they're also allowed to have cables under tension. And there's a lot of work discussing how the geometry of the structure impacts the rigidity. And so the question that you're asking is, with one of these bar hinge frameworks, you're asking, can I move the structure so that I preserve all of the lengths, right? Because it's bars and hinges. And so you're saying, can I preserve the lengths of the bars and hinges? Because if I can't, that costs energy. Okay, so let me write that out in words. A structurally rigid system is where no non-trivial global motion preserves these constraints, right? These bars are these cables, right? So the idea is, do I have a motion of my system that preserves those constraints? And if I do not, then if it costs energy, basically, to move the structure, then the structure is rigid, right? That's the idea. So if I don't preserve the constraints, I'm rigid. Okay, but it turns out that testing for structural rigidity is an NP hard problem, so not a polynomial time solvable problem, even for planar graphs. Wow, Bill Anderson is right. This is amazing. Okay, So people have come up with simpler tests for rigidity. Okay, so one simple test is called first order rigidity. And it says, okay, well, instead of testing this hard thing, maybe I can ask if there's no non-trivial global motion that preserves the constraints to first order in perturbations to the constraints. Okay, first order rigidity. And then, of course, the next thing you would think of to do, and actually it turns out this is very easy to test. I'll explain in a minute. Then you can also have second order rigidity where no non-trivial motion preserves the constraints to second order. Okay, those are the ways that this has been defined in the literature. So these are proxies that work sometimes for predicting structural rigidity, and they're much easier to test. Okay, so just again, for mathematicians and structural engineers, a floppy system is one you can push on without changing the constraints or the geometry. In our rigidity manifesto, we say, wait a second, physical materials always have an energy functional. And all of the examples I showed you today, and almost all that you can think of, are usually, at least to first order, they're quadratic in the bonds or the constraints. So they can be written. So this is the model I showed you for cells. This model is clearly quadratic in a set of constraints. So again, and this one is the one for biopolymer networks. And this is quadratic. So this is just the rest length of the spring. And this is a quadratic penalty in changing the length of that spring. You can also, if you want, add on penalties for changing the angles if you wish. So I'm just showing you that in physics, we care about the energies, and the energies are quadratic in the bonds typically. Okay, so for us, for a physicist, a floppy system is one you can push on without changing the energy. You don't care about the geometry, you care about the energy, right? Because if you can, let me sort of say the point, there may be motions that don't preserve any single constraint, but conspire in such a way to preserve the energy. Could be, right? And you should care about those, because to a physicist, if I can push on the system and the energy, it caused zero energy, then it should be floppy. And I don't care if I change some of the constraints in the process. Okay, so we want to consider that. Okay, and so the question I'll be asking for the last maybe 5-10 minutes of this talk is, when does this happen? You know, can we understand the mechanisms driving this type of rigidity? And the way we're going to think about it is we're going to add this new definition, this definition of energetic rigidity, which we say any non-trivial motion increases the energy. Okay, and now we're going to ask the question, when does first order rigidity imply energetic rigidity? And when does second order rigidity imply energetic rigidity? It turns out, as far as we can tell, no one's asked that question before. Okay, and it's very important because it's relevant for all of those examples I showed you. So that's why it's been driving me crazy for 5 years, this is the question you have to ask. Okay, so this is the question we asked. Okay, and so now I'm really going to do a little bit of math, but I'm going to go through it quickly. So the idea is that you have an energy that is quadratic in some constraint. So again, a constraint is something like, well, the difference between the length that I actually am and the rest length of my spring. That's a constraint, right? And so quadratic in the constraints just means a spring network where I have L minus L naught squared, okay? That's this. And we, more importantly, because actually it turns out the mathematicians and structural engineers don't insist on this, we insist that the system starts at a minimum of this energy, because now we can define it. Now that we have an energy functional, we insist that the system starts at a minimum of the local energy. Okay, so that means that you can prove a bunch of things because you're starting at a minimum of the energy. So a lot of things go to zero. Okay, so now you do the standard thing. So I'm going to go through this really quickly. You perturb the system coordinates, you look at the variation in second order in the perturbation, and then you insist that if I want my system to be floppy, then this variation in the energy has to be zero. So if there exists at least one perturbation where the energy is flat to second order, then we say that the system is floppy, right? That kind of makes sense. Okay, otherwise we call it energetically rigid. For those of you who think about these things, this is deeply related to the Hessian of the system. So this is the matrix of second derivatives of the total energy with respect to the particle positions. And importantly, it turns out that you can write this Hessian as a sum of two terms. There's one that is dependent on a rigidity matrix, which I will talk about more in a second. And then there's a part which we call a pre-stress matrix, because it turns out this matrix is only non-zero if the constraints are not satisfied. So the constraint, if you have springs that are unhappy, then this thing is some number, okay? And if all of the springs in your network are happy, this is zero, okay? So this is called the rigidity matrix. It's actually it's a very natural object to think about. It's just the derivatives of the constraints with respect to particle positions. It's also sometimes called a compatibility matrix or an equilibrium matrix. It's called like 16 different things because this thing shows up in every field in the world. Okay, so this is the rigidity matrix. It has M constraints. So the number of constraints are the number of rows in this system, and it has the number of degrees of freedom's columns. So it's a very natural object. Okay, and so we're writing the Hessian in this way. Okay, and so I'm not going to explain this. This is a nasty equation, but all I'm saying is that if you acquire that this matrix is basically the variations cost zero energy, then you can write it in this other way involving these matrices. Well, I'm not going to talk about it too much. So now I am going to go back to constraint counting because it turns out first order rigidity is constraint counting. So let me show you how that works. If you want, basically, so what's first order rigidity? Just to remind you, it means that you have basically, you think about whether there are perturbations, first order perturbations in the constraints that allow the system to remain floppy. Okay, and so if we have a zero mode, a first order zero mode, that basically means that I can perturb the constraints to first order and it won't change the lengths, right? I can perturb to first order and not change the lengths. And it turns out that means this is in the right null space of the rigidity matrix. Okay, if there are no first order zero modes, it's first order rigid. It turns out that those states of self-stress that I introduced a lot earlier in the talk are the states that are in the left null space of the matrix R, this rigidity matrix. And so constraint counting is just a rank nullity theorem for this rigidity matrix. So remember, this was constraint counting where we had to pay attention to the number of zero modes and the number of states of self-stress. So first order rigidity is constraint counting, and it's just true by this rank nullity, because linear algebra. Okay, so let's talk about, okay, first order rigidity works sometimes, but not all the time. So now let's see if second order rigidity works for some of our systems. So I am absolutely not going to tell you anything about this slide other than we can do second order rigidity in these systems. And it's just, if there are no second order modes, we call the system second order rigid, again, in these constraints. The important point is that you can check for second order rigidity using in polynomial time, because second order rigidity, it turns out, is equivalent to finding the root of a polynomial system. So we said generic rigidity is NP hard, second order rigidity is P hard, and then first order rigidity is real easy. You just count the constraints. Okay, so now we want to know what's the relationship between first order, second order rigidity, and this thing we really care about, which is energetic rigidity. And I'm, in a colloquium, I'm just going to sort of tell you the answer. Okay, so the question we're asking is, do these simpler things, does first order rigidity or second order rigidity imply the thing we really care about, which is energetic rigidity? And so what we've done, and this is a flowchart showing it in this archive paper, is that you can actually carefully go through a bunch of cases, and we can show formally that in some cases, first order rigidity does imply energetic rigidity. In some cases, second order rigidity implies energetic rigidity. And in some cases, you can't say anything at all analytically. Okay, so you know how I talked about spheres, like jammed spheres? It turns out that packings of sand or spheres fall into either this category, either this category or this category, which means that first order rigidity implies energetic rigidity. And otherwise, constraint counting works. Here, second order rigidity implies energetic rigidity. And so this is includes those spring network models, so extracellular matrix and vertex models, confluent tissue models. Most of them fall into this category. So what does that mean? That means that cells and fiber networks become rigid because second order perturbations to their constraints cost energy, not first order ones. So constraint counting, you only consider the first order, and that works for jammed packings in sand. And for mechanical metamaterials and vertex, for cells and fibers and metamaterials, the second order rigidity is what causes them to act rigid. And then there's some fun things you can think that actually there's a lot of subtleties, and maybe this category is very fun for thinking about materials design. Okay, I'm going to skip this slide entirely. If you're a structural engineer in this audience, I can tell you about what it means later. Okay, so what I can sort of say, I actually already said all of this. You can write pages and pages of math that I will not bore you with. And I'll just end with some sort of examples. So this is a 2D spring network. And what we knew already from numerical simulations that as you change the rest length of the spring or the box, or equivalently the size of the box, you go from floppy to rigid. And I can even show you a movie of what it looks like as you pull on the box. So we're pulling, and right there you saw that at a critical point, there was a state of self-stress actually that popped in. And then there was more and more tension along fibers in the network. And so this process we can now show formally is due to second order rigidity, to second order perturbations in the constraints, which is really exciting. And then we can also do it for these vertex models. Again, I'll just show you some pictures because videos are more fun. So this is a system that's solid-like, and you tune this tuning parameter just a bit and you get a fluid. Okay. So sort of the summary of what I've told you is that rigidity in many standard materials is described by first order rigidity or constraint counting. That includes things like sand, okay? Rigidity in biological tissues, including both extracellular matrix and the cellularized tissues, is described by this new concept of energetic rigidity. And in these cases, second order rigidity is a good proxy. So second order rigidity explains the rigidity in those tissues. In general, metamaterials that are rigidified by higher order rigidity don't require you to change the topology of the network in order to get rigidity. So you don't have to crowd them or change the number of contacts or packing fraction. You can just change a simple tuning parameter. And so the onset of rigidity occurs when a state of self-stress appears due to very small changes in a parameter. So that leads to the following outlook, which is, biological tissues really are metamaterials that are rigidified by these terms beyond first order. And you can start to think, well, this could be a very exciting design principle for bio-inspired materials. You can ask, well, maybe we could start with this theory to think about the space of possible designs and think about what biology might take advantage of. So I won't show this whole movie again, but clearly the Terminator 2 is a good example. Okay. So thanks so much for your attention, and I'm happy to take any questions. Oh, and up here behind here, this is Syracuse University where I work. And so I'd be delighted if any of you in the audience, especially undergrads or graduate students, want to talk to me about what it's like to be there or, opportunities at Syracuse.
So, thank you very much, Lisa. Thank you very much for this wonderful talk. Are there any questions? Alberto, can you see the questions, or where is the chat? Let me see the chat. Are there any questions? No questions.
Eric Vasquez.
Eric Vasquez. Okay, go on. Eric.
No, I didn't raise my hand, but I was actually thinking to ask something, so I guess somebody was reading my mind. So I'd like to. I was wondering now about these metamaterials. How I guess I was just trying to understand some gradients in the temperature. How easy is to study that? Because I suppose that, I mean, maybe a couple of degrees or so, it's not important, but how can that be modeled and how can be understood? I mean, that's something interesting, right? You have a gradient in temperature here associated. I didn't have.
Yeah, so I think so there's a couple of questions there. First, I didn't talk very much about how thermal fluctuations, just even a bath of not having a gradient in thermal fluctuations affects these systems. So I could talk about that a bit. And then, I mean, I think thinking about how a gradient in temperature might be used to actuate these systems is another interesting question. So I'm not exactly sure which one you're asking or if I should try to answer both.
Yeah, I was asking about the first one, but both would be great. Yeah.
Okay, so the first one is that yeah, it's quite often, right, that if you take, well, I mean, we're most familiar with this idea from quantum critical systems, but if you have a critical point and then you turn on a temperature or fluctuations, right, you often see that critical point expand into sort of a critical regime at finite fluctuations. And so that really does seem to happen in these materials that have this rigidity critical point. That's a second order critical point, second order rigidity critical point. It also turns out that quite formally, the transition in these models is a second order critical point that's in the Ising model universality class with like the same, it has mean field exponents and stuff, but that's a different story. But when you add these temperatures, what you're doing is you're sort of sometimes stiffening, you're adding fluctuations to the system, which can in sometimes some ways stiffen parts of the system. So, because you're sort of at any given time, like if you imagine a spring, you're sort of stretching it out a little bit extra with that thermal fluctuation, and that makes it more tense, right? But you also are introducing sort of the possibility to sort of induce rearrangements of the topology of the network. And so what that does is it takes that sort of one critical point and broadens them out. And so you have a whole bunch of interesting behavior. So basically you can see that critical point and sort of critical behavior over a wide range of phase space at finite temperatures. Also, as you know, from just like normal solids, you can increase the temperature enough to make all of these systems floppy. So there's another sort of phase. So if you imagine a temperature axis like this, and here's your tuning parameter axis, such as the rest length of the spring. If you turn on the temperature high enough, there's a transition. You can go from solid-like to fluid-like if you just increase the temperature far enough. Okay, and then gradients in temperature are a really interesting way of then actuating systems, because of course we're envisioning making systems where you have low-lying, low-frequency normal modes, right? That would be an interesting way to like displace this whole structure. And the fact that you can do it by just tuning a small parameter is very interesting, because then you can think, well, maybe one way of effectively tuning that parameter is just having a temperature gradient. So you could think about changing the effective rest length of the spring by putting one part of your system under a slightly higher temperature than another, and that would give you these types of behaviors.
Okay. Yeah, this came to me because when I saw the movie, the Terminator movie, and I just see the fluid moving, that always reminds me about the Leidenfrost effect, right?
Oh yeah.
So that reminded me that the size of these fluids, the size of basically each the size of each drips, it's determined and has some gradient in the temperature. So that's why I was thinking about this gradient, right? Okay, Thank you.
Thanks.
I'm sorry. We have another question by Ruben Barrera. Please go on.
Well, first, it was very nice, very nice colloquium, very nice talk. I was very happy to be here. Also, I was wondering if you have thought about the propagation of waves in this type of systems, in the sense that it could be more or less easily to measure or to check more of your Lagrangian or of your energy parameters. So I was wondering that if you have thought about it, if you have some work, or if you think it's interesting.
Yes. So the short answer is yes, it's very interesting. So from a computational side, you can ask a question such as, what is the vibrational density of states of these systems,
Sure.
for example? And they have
Exactly,
really
sure.
interesting densities of states. So I also study vibrational densities of states in glasses as well, just like normal standard glasses. So those already have very interesting properties, such as like quasi-localization of excitations and two-level systems that present interesting things to the propagation of sound, and they have interesting heat capacities. So I would say that these systems, because they're disordered, share many of those same features that you see in glasses. So they also have a population of two-level systems. They also have localized excitations at low frequencies, but there's something crazy about them. And the crazy thing, I think I can sum it up most, and I don't understand it. So if anybody's interested in working on it, it's an interesting problem to me, is that normally, as you think about changing the temperature in the system, the effective energy barriers in the system go up in these disordered systems. It's called super Arrhenius behavior. It basically means that the effective energy barrier, so you're exploring a complex potential energy landscape, and as you go to lower and lower temperatures, the effective barriers you have to cross get higher and higher, which makes perfect sense, right? And in these systems, in these models, the exact opposite happens. They're sub-Arrhenius. So like you go to lower and lower temperatures and they actually get easier. It gets easier to cross barriers. They let, and also, and since we're, you asked about a harmonic question, which is, they also get softer harmonically. So like the potential wells get softer and softer at lower temperatures, which I've never seen before. So I don't know.
And also,
okay.
I was wondering if it's possible to take account of dissipation. So some of these systems might have dissipation, and sometimes they may rule the mechanics or the motion of the dynamics of the system. So I was wondering about usually in this type of formulations to put on dissipation is not a simple task, but I wanted to ask you.
Yeah, so that's a great question. So a couple things. The first thing is that yes, these systems often have a lot of dissipation, and actually, when we simulate them, it actually let me see if I can find the slide where I was showing this. This system is actually, we put in to do this simulation here. Let me press play. We actually have put in a lot of dissipation, and in fact, we're in the overdamped limit, so that the system is like, when you solve the equation of motion, you're basically solving F equals V instead of F equals ma. So this system, as we simulate it for predicting the behavior of like cellularized tissues, there would be it would all be diffusive propagation. You'd get no sort of inertial propagation in the system.
Sure.
Nevertheless, the sort of vibrational density of states really, it turns out, tells you a lot about the collective dynamics that you see in the system, because it turns out that in active matter, so these are all active systems where they're self-propelled, right? The cells are self-propelled. And it turns out that in active systems, unlike equilibrium systems, the dynamics sit right on top of the low frequency modes in the system. So even though you might have thought that solving for the low frequency modes was boring in a system that was diffusive, it turns out that this system actually, because it's active and activity kind of acts like inertia in some ways, it actually is worthwhile to study the vibrational density of states, because even though sounds don't propagate, the activity makes things look like sound waves.
Made sense. Well, Thank you
Yeah.
very much. It was,
Yeah.
thank you.
We have another question by Giuseppe Piruccio. Giuseppe.
Hi, nice talk, thank you. So I have two questions. The first one is, if I understand correctly, all the things you show, basically the system is isotropic and kind of homogeneous. So what happens, can I think about something anisotropic in which if I apply the stress or the force in one direction, I see a rigid body, and if I apply it in the second one, a different direction, I see a floppy one. Does it exist?
Yeah, so so the first question is that this system, this fruit fly system I talked about, is actually highly anisotropic in exactly the way that you state. So it turns out that fruit flies have this crazy biochemical signaling pathway where they line up all the tensions on the vertical interfaces, and so it smooshes all of the cells like this, you know. So you start out with an isotropic system and it smushes them first. And so it turns out you can show that by looking at a nematic order parameter, like a cell alignment parameter, in addition to the cell shapes, you can sort of figure out in these anisotropic systems how the rigidity changes. So it turns out that sort of what you're actually looking at here, if you don't mind me taking a second to tell you,
Sure.
is if I had an isotropic system,
Yeah.
then basically I would expect no cell alignment. You can prove that. It's not obvious, but you would expect in systems that don't have other aligning interactions, you don't get alignment. And so this axis is a fluid solid transition that you would expect in the absence of anisotropic forces. And that happens at a certain value of the shape index. And then as you add more and more forces, it turns out that is sort of more anisotropy. So let's say you were at a fixed value of the cell shape. So the cells weren't changing their internal shapes that they wanted to have. And you just turned up basically added more anisotropic stress. That would be going along this axis, and you could actually turn the thing into a solid, exactly, I think, as you said. Okay. And then if you put it I mean, we didn't show this here, and actually it's kind of interesting to do, but if you apply a stress in the opposite direction to change it back to isotropic, it would go back this way and become floppy again. So this paper and some of the supplemental materials kind of has all of the information about how you think about that.
Anyway, the two phases do not coexist, never. It is always either.
Yeah, that's actually really okay. So, because you started out by asking also about heterogeneity, and then you didn't ask me, but you said it. And so, if you have a mixture of cell types where you have cells that want to have different shapes, then you can have coexistence. And you can ask fun questions like, do they spontaneously segregate and sort, or do they stay mixed if you have sort of two phases? And yeah, we've done a lot of work on that. I don't want to bore everyone with all of the details, but yeah, there's a lot of interesting questions there.
And the other question was on a different topic. Is there anything like a non-Newtonian fluid somewhere here? I mean, something that depends on the way you act on the fluid, and then it reacts in a solid way or rigid way or in a fluid way. Is there anything like this?
Yeah, so the fluid-like phase of this is actually, I mean, I call it a fluid, but it's not a Newtonian fluid. It's viscoelastic for sure. So we've done some work actually probing the effective rheology of these types of systems, because when I say fluid, I just mean in the limit that you go to a time scale goes towards infinity, is it fluid or not? And it is, but there's a short time scale on when it's solid like, and it looks a lot, I don't know how much, like it's a lot like a Binghamton plastic actually rheology in these models.
Okay, thanks.
Okay, I would like to thank you, but before just let me ask you a final question, because I would like to know your opinion about this. Because when we go to the undergrad classes of thermodynamics, we learn about this fluid and solid transition that you have here. However, usually the books don't say anything about rigidity. You know, and one of the paradigms of phase transition is that you have a broken symmetry and a Bohr under parameter. And well, you were a student of Langer. I think he has a book on that or something.
Yeah.
So I was wondering, what's your opinion about this? What is the order parameter related with rigidity? Why do books never study fluid solid transition, talking about rigidity, which is the most important thing that defines what is a solid and what is a fluid?
Yeah. Yeah, it's good. I can think of like 3 different answers to your question. So I agree. I mean, I think that we should teach Goldstone modes to undergraduates. So why? Because so I think that this is, so the simplest answer for me sort of to say is, well, anytime, I mean, and this is a little hand wavy, but it's hand wavy at, I think, a good undergraduate level. And I think it has the right I mean, like, it's like sort of morally correct, which is that, okay, anytime you have rigidity, you really do have a broken translational symmetry, in the sense that, like, these because like for any material, you have some, it might be liquid-like order, but like you have a G of R. So you predict that if I have a particle here, there is no particle here. It has to be a certain distance away before I see it again. So in that sense, you really have a broken translational symmetry. And therefore, all of these, everything I showed you has phonons, right? In the sense that if I have a broken, sort of a spontaneously broken translational symmetry, then there's a Goldstone mode associated with it, which is just the long wavelength perturbation of that symmetry. And the long wavelength perturbation of a broken translational symmetry, right? I have my ruler, I shift it here. The long wavelength perturbation of that is a longitudinal sound wave, means that I sort of perturb it a little bit here and then not much. And that, that is the Goldstone mode associated with that broken symmetry. And I agree, we don't teach it that way, but I think hand wavily you could, and that would be a good thing to do. So whenever I teach solid state physics, I teach it that way.
So thank you very much, Lisa, for this wonderful talk. And we thank again, Lisa, and we hope to see you in Mexico in person.
That would be lovely, actually. Thanks so much for letting me talk anyway. It was nice to see you. I'm sorry I didn't see all of your faces. I know we had to turn off cameras because of bandwidth issues. And I never know if I give a good talk if I can't see anyone's faces. So hopefully it wasn't too bad.
Maybe someone can open their cameras to show Lisa how the Mexican students looks like.
Yeah, if you want, I would love just to wave at you and say hi. So I feel like
Please say hi to Lisa.
At least my you're welcome to turn on your camera for a moment if you want, I think now, but you don't have to if you don't want to. But it was nice to see all of you, and thank you for listening to my talk.
Thank you, Lisa.
Okay. Well, bye-bye, Lisa.
Thank you very much for your talk.
Yeah.
Thank you.
My pleasure. Thank you.
Bye.
Bye.
Thank you so much. It was so much fun. Thanks.
Thank you. All right. Shall I duck off now? Is there for those of you who are organizing, is there anything else that I should do right now?
No, I think it's okay. It's okay.
Okay.
Thank you very much, Lisa. Yeah. Nice to see you again.
Yeah, it's nice to see you too.
Bye.
Bye.